This is Bhavani, lecturer from Department of Physiology from SRM Dental College, Ramapuram. So today I am going to teach about compound light microscope. So first we can know about what is microscope. So why we are going to use a microscope, why a microscope is needed for us. So microscope, from the meaning itself, micro, so micro means small and scope means view. So this has been found out by a Dutch scientist, Antony van Leeuwenhoek. He has invented the microscope in 17th century. So before that, many scientists has undergone many invention related to microscope. So why he has found out the microscope is to study the microcells, the cells which we cannot see through our naked eyes. So those cells, to find out those cells, he has found out this, he has invented the microscope. So we are going to see about a compound microscope. So compound microscope, we have two types of compound microscope. So one microscope we call it as your compound microscope, the other one is your compound light microscope. So then the pillar which stands with the coarse adjustment and fine adjustment, and you have a handle to carry the microscope and a body tube where you can find your eyepiece, then revolving nose piece we can find, where you can find different kinds of lenses which is used for your magnifications. Then the mechanical stage where we are going to place a slide. So why we call that as a mechanical stage? Because it has the screws, mechanical screws, which can move the slide either forward or backward or in your sideways, right side and your left hand side. So that's why we call this stage as your mechanical stage. Why? Because it has the screws in them. So next is your illumination system. So illumination system is your light source which is present in our microscope. So we have a light source, so the light color, the bulb color will be yellow in color. Then we can find a condenser. So why we need a condenser is to condense the light and it sends to your slide, it hits up your slide. Then you have a iris diaphragm. So this iris diaphragm is there to, why it is present is because to the amount of light rays which should enter into a slide. So for that you have an iris diaphragm. So all these portions are present at your base of your microscope. So near your light source you can find a knob which is used to adjust your light source either to a low power or a high power. So this we call it as your illumination system. Next is magnification system. So here this magnification is going to be done with the help of your eyepiece and your objective lens. Okay, so we are going to view through the eyepiece with the help of your objective lens. We are going to see the slide. So in your objective lenses you have four different x's. So one is your 5x, 10x, then 45x and 100x. So the eyepiece can be either 5x or 10x. So here the magnification is going to be done between these two lenses, and the cells which are present in the slide is going to be magnified and we are going to view through your microscope. So here the magnification is the multiplication of your lenses. So the eyepiece lens is 10x and your objective lens, when you place it in a 5x, the 10 into 5, which is 50 times the cell is going to be magnified. When we are placing your objective lens as 10x, the 10 into 10, which is 100 times the cell is going to be magnified. Next is 45x. When you place your objective lens in 45x, so 10 into 45, so where 450 times it is going to be magnified. So when we are going to go for an 100x, this is going to be magnified for 1000 times. Here when we come to your objective lenses, the 100x, so when we are going for a higher magnification, so we need to add up a oil immersion. So we'll call it as an oil immersion. So in 100x we have an oil immersion. So the next is your adjustment system. So adjustment system is your final one. So where you are going to adjust your cells to view clearly. So where you have two kinds of adjustments. One is coarse adjustment, another one is your fine adjustment. So coarse adjustment is present. Why? Because to get a clear picture of where the image is present. Okay, so this we call it as your coarse adjustment, and fine adjustment to get a fine picture. Okay, the clear and a fine picture. So you have your fine adjustment to see a proper color image. So this is, we call it as your adjustment system. So these are the parts of a microscope. So thank you.